Hello, everyone. Welcome to my video tutorial for single-cell immune receptor analysis. Today, I'm going to show you how to use the Scirpy package to analyze the single-cell immune receptor in Python. In the first part of my video tutorial, I'm going to show you how to import receptor data into Scirpy. First, let's have a look at the online tutorial for the Scirpy. You can see here is the online tutorial for importing receptor data into Scirpy. So depends on what kind of data you have. They developed different functions to load the data into Scirpy. For example, we have the read 10 times VDJ function to read data from 10 times genomics cell ranger output. So for today's demonstration, we are going to use this function. If you have data analyzed by other packages, for example, from TracerR, then you can use read tracer function. From BracerR, you can use read bracer function. So all the functions for loading data were listed here. You can select one to load your data according to your data format. As I mentioned, we are going to use the read 10 times VDJ function. So the data set that I'm going to use for today's demonstration is the same data set that was used for the Scirpy demonstration. The data set is CSE 139555. You can see in this data set they performed both single-cell RNA sequencing and also analysis for the T-cell receptor clone type in cancer patients. In this data set, they have three sample types: the tumor, the normal adjacent tissue, and the peripheral blood. So they did the sequencing from 14 patients. And the data is available for you to download. You can see down here. They analyze the data in R, so they have the RDS data available for you to download. You can see the all integrated data, metadata, then CD45 integrated data, and the T-cell integrated data. Because their data saved in RDS, so for today's demonstration, we are going to download the original data. You can see here they have 32 sequencing dataset. We are going to download the first one for demonstration. So you can see the data were analyzed by 10 times genomics cell ranger software. You can see the T-cell sequencing data, quantic annotation CSV for this dataset, and also you can see the barcodes, genes, and the metrics for the single-cell RNA sequencing data. So we can tickle the data for the first set and download for today's demonstration. If you go to the end, you click download, you will download the data. So I downloaded it already. You can see in my folder, we have the filtered quantic annotation CSV file for the T-cell sequencing information. Then in the IT1 folder, we have the barcodes, genes, and the matrix data for the single-cell RNA sequencing data. So after you download the data, you need to unzip the data and also change the file's name to just the barcode, genes, and the matrix. Then we can load them into Python for today's analysis. So we have the data here. Now we can go to Python and perform the analysis. So you can see here is the code we are going to use for today's analysis. First, we need to load all the packages that are required for today's analysis. Let's run. Okay, we loaded all the packages. Next, we can use the read 10 times VDJ function to load the TCR sequencing data. You can see here is the files path that I save the data. We can load the TCR sequencing data and name it as A data TCR. Let's load it. So we can have a look at the data structure. We can just run the name data TCR. You can see we have 6,784 sequence information for the TCR sequencing analysis. In the dataset, we have the unstructured layer named as Scirpy version, and importantly, in the observation multiple layers, we know this is an array immune receptor repertoire dataset. This is a dataset lately developed using word array to save the TCR sequencing data. So next we can load the single-cell RNA sequencing data. We can use the read 10 times mtx function. This is the function that we used in Scirpy to load the single-cell RNA sequencing data from 10 times genomics sequencing data. Again, we need to specify the files directory. I showed you. I saved it in the ITL folder. Then we use the variable names as gene symbols. So we can load the data as A data. At the same time, we make the variable names unique in case there are repeated gene names. Let's load the sequencing data now. If we have a look at the A data, you can see we have 7,462 cells here, and we have 30,727 genes. So recently they updated the Scirpy package to incorporate the MuData function. MuData is a Python package that you can analyze multiple models. So we can combine the single-cell RNA sequencing data with the TCR sequencing data as M data. If you use the older Scirpy package, they save the TCR sequencing data information in the A data observations. But now we saved the TCR sequencing information as a separate data array immune receptor repertoire. So let's combine both datasets as M data. If we have a look at the M data now, you can see we have two modalities. CX is the single-cell RNA sequencing data. Array immune receptor repertoire is the T-cell sequencing data. You can see in total, we have 7,603 observations. But in the single-cell RNA sequencing data, we only have 7,462 cells. And in the TCR sequencing data, we have 6,784 observations. You can see the observations number in the M data is bigger than both datasets, which means some TCR sequencing data they have the cell barcode that are not presented in the single-cell RNA sequencing data. So we have the new data now. First, we need to analyze the single-cell RNA sequencing data to cluster the cells. So we can filter the single RNA sequencing data to keep genes that expressed at least in 10 cells, and keep cells that have at least 100 gene expression. You can change this setting according to the quality of your dataset. So we can filter the single-cell RNA sequencing data now. If we have a look at the data now, you can see we only filter out one cells. Then we filtered out 17,025 genes. You can see here we only have 13,702 genes left. In the beginning, we have 30,727 genes. Because we are analyzing the single-cell RNA sequencing data, no changes for the T-cell sequencing data. So here we only performed the filtration by cell numbers and gene numbers. If you know well about the scMP package, you can also plot the variant plot and subset the data according to the total count in each cell and the gene numbers in each cell. In scMP quality control, we also calculate the percentage of mitochondria genes, but in this dataset, if you look at the genes in the gene files, we don't have mitochondria gene information. So you don't need to perform the mitochondria percentage analysis. So after the data filtration, we can run scMP workflow. First normalize the cells, then logarithmize the data. After that, we can find the highly variable genes. Then we scale the data, run PCA, find the neighbors, run UMAP. And in the last step in this workflow, we are going to use the lead method to identify the cell canisters. So let's run the scMP workflow to analyze the single-cell RNA sequencing data. You can see it is running. Okay, it is done because we only downloaded one dataset. It is very quick. In the original paper, they analyzed all the data, and they said in R, it requires at least 113 GB computer memory. So my computer will not be able to do that. That's the reason we only downloaded one dataset for the demonstration. So after the standard scMP workflow, we can have a look at the observations in the single-cell RNA sequencing dataset. You can see now we have a new metadata column named as latent. Here are the latent cell canisters after the scMP analysis. So we can plot the cell canisters, use the scMP plot UMAP function. We specify the data will be the single-cell RNA sequencing data in the M-data. Then we can count the cells by latent canisters. At the same time, we count the cells by CD3E expression. This is a mark gene for T cells. Let's plot both figures. You can see down here, we have 14 cell canisters from this demonstration dataset. Then you can see almost all the cells express CD3E gene. Because the cells were purified by flow cytometry using the CD3E antibody, so we analyzed the single-cell RNA sequencing data and we clustered the cells. So next we can start to analyze the T-cell sequencing data. First we can create the chain index, use the index chain function. If we run this function, you can see it is filtering the chains. In this dataset, we have the VJ chain and the VDJ chain. We don't have other chains for the T-cell receptors. So we created an index for the VJ chain and the VDJ chains. Next, we can perform the TCR quantity control using the chain quantity control function in the Scirpy package. Let's run this function. You will see what kind of quantity control we are going to perform for this dataset. Let's run. You can see through the chain quantity control, it analyzes the receptor types, receptor subtypes, and the chain pairing. We know in this dataset we only have the TCR receptor type. The receptor subtype means the clone have different DNA sequence, but they have the same amino acid sequence. So they can recognize the same antigen. So the third quantity control is the chain pairing. We will analyze the receptor type, receptor subtype, and the chain pairing in my second video tutorial. So now we analyze both single-cell RNA sequencing data and also the TCR sequencing data. We can plot the data to see the TCR receptor information in the cells that are analyzed by single-cell RNA sequencing data. So first we can plot the Leiden canisters. I showed you already we have 14 cell canisters in this dataset. Then in the second figure, we count the cells as receptor types. You can also use receptor subtypes and chain pairing to count the cells. Let's run to make the figures. You can see we generated two figures. The one on the left-hand side is the Leiden canisters. We have 14 cell canisters here. You can see on the right-hand side, the cells were counted by TCR expression. You can see majority of cells express the TCR receptor. They were counted in blue. Then some cells show the multi-chain. Those cells will be the double knit or multiple knit. Then the cells don't have TCR receptors were counted in green. And also some cells were labeled in gray color. I showed you in the beginning, the mu data observations is bigger than both single-cell RNA sequencing data and the TCR sequencing data. The cells were counted in gray were only presented in one dataset. Okay, I used the one dataset as the demonstration dataset from the GSE 13955 dataset. I showed you how to load both single-cell RNA sequencing data and TCR sequencing data and combined it into M data. Then we analyzed both single-cell RNA sequencing data and TCR sequencing data. So you can use this method to load your data and perform the T-cell clone type analysis. Okay, that's for today's demonstration to show you how to import data into Scirpy and create M data for clone type analysis. In my next video tutorial, I'm going to show you how to analyze the clone types using the Scirpy demonstration dataset. Because in this video, we only loaded one dataset, but in the original dataset, they have samples from 3 different tissues, the tumor, the normal adjacent tissue, and the blood samples. So in the Scirpy demonstration data, they actually created a demonstration dataset containing 3,000 cells, but those cells have different origin from tumor tissue, normal adjacent tissue, and blood samples. So in next video tutorial, we are going to use the demonstration data. The demonstration dataset was saved in the Scirpy package. So you can directly load the demonstration data from the Scirpy package as mdata. If we load it and have a look at the mdata, you can see in this dataset we have 3,000 cells. For the single cell RNA sequencing data, we have 30,727 genes. You can see for the TCR sequencing information, we also have 3,000 rows. So I'm going to stop from here. I hope to see you in my next video tutorial for the clone type analysis. So please subscribe my channel if you haven't to do so, and share my videos with your friends. Thank you.